everybody. So there's a slight switch of plans at the end, and I'll be filling in for Dr. Alfano. My name is Marcus Curl, Director of Product Applications with InVitria. Before we go to lunch, I want to introduce InVitria, the product that we provide, the problems we're trying to solve, and how you can implement these solutions today. So the mission at InVitria is really to future-proof our media supply. And the way that we do this is we focus on recombinant serum proteins to replace serum throughout the entire manufacturing process. When we look at the therapeutics that we're working on and the patient populations that these can serve, we also have to take a look at our raw materials and the media that we're using and the proteins in these media that really give them their function. And how do you create an environment that will scale to ultimately meet the needs of these patient populations? And so our goal is to completely decouple manufacturing of gene and cell therapy from serum and from the serum market and provide a recombinant alternative that's chemically defined, a term that I call blood-free, which I'll go into more detail here shortly, and ultimately create a safer therapeutic. And another one of our goals at InVitria is to really help clarify what's meant by the different categories of media that you have to work with as you look to large-scale manufacturing of cell and gene therapy, vaccine manufacturing, where you're sourcing serum for these different processes. And we've created a new category to kind of help differentiate, known as blood-free, and I'll go into a little more detail here in a second. So quickly about InVitria. Again, our core competency is recombinant protein production. We specialize in serum proteins such as recombinant albumin, recombinant transferrin. We have a few other insulin transferrin supplements, as well as complete media. And our goal is, if you look at the cell types we're expanding and the conditions they're expanded in, how do you replace the proteinaceous components with safer, higher performance alternatives that ultimately lead to more control of your production system and a safer final product for your end user? And so InVitria, again, specializes in creating what we call blood-free media. And this is a step above, in my mind, xeno-free and serum-free, which, while their messaging seems like it wouldn't contain whole serum or wouldn't contain serum proteins, either contain bovine serum albumin in the case of serum-free media, or human serum albumin or human serum transferrin in the case of xeno-free media. And so what we've done is we found a way to make these recombinant proteins in a scalable way that can meet the needs of any manufacturing process while maintaining the same performance, but improve on consistency and improve on the safety, ultimately, of your manufacturing platform. And so a quick overview of the company. We have two main locations here in the United States. In Kansas, we have our complete manufacturing, so gene to protein, as well as our complete media formulations. Again, these are manufactured in animal-free environments at the tertiary level. So our expression system, the equipment we use, the final packaging, all has to be completely free, or in the history of the use of that equipment, never touch animal components. Again, the goal is, how do you, quality by design, take away the adventitious agent risks that can come from mammalian-derived components? And so from the manufacturing itself, we've kind of built in that safety as well as that control. In Colorado, we have our product development lab. So this is where we work with different cell types, different types of applications, different types of buffers, final formulations, to develop data and to develop formulations that can help our customers in specific applications to replace these serum-derived components. And again, the culture work we do there, we have to do it in Colorado because in Kansas we can't have mammalian cells on that campus. So we have to do all of the work and development for the media formulations that are then manufactured in Kansas and Colorado. And some of the values we're trying to bring our customers, again, in addition to thinking about the future, thinking 5 years ahead, once your therapeutic is approved, how do you source the raw materials you need? Also increase the performance and consistency of your system. Through chemically defining your system to the utmost level where even your proteinaceous components are all recombinant, you can use the media as a tool. And what I mean by that is by understanding the metabolic demands of your specific cell type, by understanding the stresses and the pressures of your manufacturing platform, you can use a media that's tailored specific to those needs. But again, the only way that it's really possible to create a consistent and manipulatable system is to have complete chemical definition. And so by replacing those proteinaceous components with recombinant, we're really able to achieve that. And a great example of this is, through some of our work in T-cell therapy and some of our research in T-cell therapy, where identifying specific phenotypes with specific patient outcomes, you have very different results. So if you're able to use the metabolism of the media to drive a phenotype that's associated with a very positive in vivo result, you can further improve your process, further improve the control of your process, and ultimately the efficacy of your therapeutic. We have faster time to market in terms of a regulatory standpoint. Again, BSE, TSE free statements, that's all we do. And the FDA, EMEA have adopted some of our solutions into marketed therapeutics. One good example that's made public, thankful to Merck, is the Merck Ebola vaccine, which includes our recombinant albumin, exbumin, in the final formulation of the actual injectable. And that's been in hundreds of thousands of patients and was designed to replace human serum albumin in that final formulation. And then ultimately, we're CGMP certified and can provide GMP media, GMP reagents at any scale. So to briefly conclude and introduce a few of our products, specifically what's available today, kind of how you can implement these benefits now, we have individual cell culture reagents and components, as well as complete cell culture media. So in terms of components, we have recombinant albumin. We have quite a few different versions of this recombinant albumin. We have exbumin for excipients, cell stemesgrate for cell culture, as well as optibumin, which is the highest reactivity albumin on the market, perfect for albumin drug conjugation, and really anywhere human serum albumin or bovine serum albumin is used, we have a solution where from a component standpoint, you can replace it with a higher performing and safer alternative. Recombinant transferrin works great in cell culture. We've also been doing some interesting research looking at leveraging the transferrin receptor during transfection as a way of complexing PEI, transferrin, DNA, and delivering it to the cell with some really great improvements. And then we also have complete media. So we have some off-the-shelf solutions that are currently used in clinical manufacturing. We have media specific for T cells, HEK293, Vero, and then we also have a very prolific custom media program. And this kind of gets into my earlier point where we want to take a tailored approach to everything about your process. Not only do we want to prove it from a supply chain and safety standpoint, but we want to create a tailored solution that's designed for your manufacturing process that optimizes your manufacturing and ultimately the performance and safety of the final product. And with that, I really appreciate your time and your attention. If you have any questions, we have a booth just around the corner. I'm happy to share some data on the relevant cell types we work in, some of the proteins we manufacture, and how we might be able to help you. Thank you. And I don't know if there's any questions, but I know it's lunchtime, so I don't blame you. All right, thank you very much.